All right, I guess it's 2:31. We can maybe get going. Yeah, so I'll start with an introduction. So first of all, before the introduction, a big congratulations to Woody and his team, and he might talk about a little about it today. But formally speaking, as a formal introduction, Woody has been a thought leader in molecular simulations and computer-aided drug design, with over 80 publications covering novel methods and applications. He received his BS in physical chemistry from the University of California at Santa Barbara, where he studied nonlinear optical properties of organic polymers using computational quantum mechanics methods. He's a reviewer for many top journals related to computational chemistry and drug design, and is on the editorial board of both Chemical Biology and Drug Design and the Journal of Chemical Information and Modeling. So, Woody, pleasure to see you again, and really looking forward to your talk.
Great, thank you. Let me share my screen here and go to full screen, and maybe you could just let me know if you're seeing the presentation mode or the presenter mode.
Presentation mode.
Excellent.
Yeah,
Super.
we see it.
Great. Well, thank you for the introduction, and thank you for everybody joining. And happy to tell you about our physics-driven approach to discover our clinical compound, SNX281, which is a small molecule STING agonist that recently entered the clinic. So first, a little bit about the company. This is a bit of the spectrum that we cover, from the supercomputer on the left all the way through the clinic on the right. We like to say we're the first chip-to-clinic company. On the supercomputer side, we have about 400 GPUs and growing, a few thousand CPUs. In our internal cluster, we use the cloud. We're doing custom hardware development as well. On the simulation platform, we have great strengths in quantum mechanics and molecular dynamics simulation, and combine those in order to understand how biological systems work in terms of thermodynamics. And then we pull together a team of scientific experts, including both experimentalists and computationalists, and integrate that into our drug discovery operation, where we have biology, chemistry, biophysics, and essentially everything that you would normally have in a drug discovery organization, but combining that with our physics-based approach and very specifically integrating biophysical data and other experimental data into our simulation methods. We develop tools very specifically for particular problems that are bottlenecks in our drug discovery. So that is our number one focus, is developing tools, whatever that happens to be, computational or experimental, to drug the most challenging targets and bring assets to the clinic. And that's what we did with our STING agonist, which I'll talk about. And we do have a deep and differentiated pipeline as well. Our labs are based in the Seaport area, right across from the Convention Center, the Boston Seaport area, I should say, on D Street. We just renovated and have about 10,000 square feet of lab space, in addition to about that same amount of office space. Everybody's together on the same floor, so we get the experimentalists and the computationalists really fully integrated, working together, and right in the Seaport area, where really a lot is happening. If you're not familiar with the Boston area or haven't been for a while, check it out online, and then you'll see it's really been going really well down there. And in addition to all the commercial stuff, there's a great expansion in the life sciences happening now as well, and we're really right there at the beginning. The news that Jabanu alluded to is that we are now part of Roivant Sciences. So if you check out our website or over the news, you'll be able to learn a lot more about that. Really phenomenal opportunity for us. Roivant are leaders in making clinical trials much more efficient, and they've taken a number of programs into and through the clinic and have approved drugs. They're very strong on the clinical and downstream side of things, and we're really part of their additional move into early discovery and create their own internal engine to feed the clinical pipeline. You can learn more about that. Again, this is from our website, but there's other news and reports and such, and happy to talk about that with anybody anytime. But to the science and what we do here at Silicon Therapeutics. So I mentioned that we have a fully integrated lab, and we do all of our experimental work as others do in drug discovery. But we bring this unique advantage in terms of physics-based simulations. And so in this movie, it'll give you a little bit of an idea of what that means. We run all atom molecular dynamics simulations, so we try to reproduce the biologically relevant system as accurately as possible with waters, ions, cofactors, et cetera. stripping away the water so we could see the protein. And what we really care about is how do molecules bind to proteins, and how does that binding modulate the function of the protein? That sometimes is through direct inhibition, but often through conformational changes. We try to understand the thermodynamics and kinetics of the system in order to make rational design modifications to modulate the conformational state of the protein, and then iterate with experimental approaches to understand how those conformational changes impact the biological function. And based on that, we drive our products and projects forward. The reason that we're running molecular dynamics simulations and the reason that this is becoming so powerful in recent years is because we can now start to access biological timescales with molecular dynamics simulations. Molecular dynamics in general is not something new to the research field. The methods have been around for decades, and there's been a lot of progress and a lot of groundbreaking academic work that's gone on in the molecular dynamics space and quantum mechanics space. But up until some recent technology advancements, it's really been inaccessible to run these types of calculations at the scale that's needed and with the turnaround time that's needed to impact drug discovery and see the biologically relevant motions. And so as you can see here in this box, a lot of biologically relevant motions happen kind of in the nanosecond to millisecond time scale. Our molecular dynamics simulations, the way we run them is we start with femtosecond time steps, so 1,000 times faster than a picosecond. So femtosecond time steps, we compute all the forces between all the atoms in the system, and then we update the coordinates based on those forces, and we do that again and again and again. And so that movie that you saw previously there was the culmination of that, where if we do 1 million of those steps, we get to a nanosecond. And if we do 1 billion, we get to a microsecond. If we do 1 trillion, we get to a millisecond. So it's very computationally intensive. And until GPUs really in cloud computing, we could only really access picoseconds of simulation time on a daily basis, which allowed us to get access to interesting things like water thermodynamics, very important for drug discovery, but not the whole story. And so now that we can run these microsecond timescale simulations, we could start talking about agonism, other conformationally related events, allosteric binding, more generally just binding energies, loop dynamics, domain movements, and things along these lines. So we can finally start to use the simulations at a scale to access timescales that really matter for drug discovery. Now, a lot has gone into building the platform of the company. I'm not going to talk about the platform itself here, but I will just mention a few of the key advantages that we've developed. We have a leading force field where we run quantum mechanics on all small molecules before we attempt to do any free energy simulations or molecular dynamics. It's extremely important to have an accurate force field, and we parameterize everything from scratch with a bespoke force field that's generated directly based on quantum mechanical simulations. We've optimized our methodology for alchemical binding free energy calculations, both relative energies between related molecules and absolute energies for unrelated molecules. We've developed a bunch of tools around water thermodynamics, atomic decomposition, advanced virtual screening methods, enhanced sampling simulations, and the integration of biophysical data. So I'm going to talk about a number of those approaches without getting into the methodological background and explain how we were able to design a novel first-in-class small molecule agonist of STING. So here's a picture of STING. In blue is the open conformation. This is the inactive conformation. And in purple is a closed conformation, active conformation that is induced upon binding to the endogenous cyclic dinucleotide ligands. What makes this such a challenging problem from a drug discovery perspective is the fact that it binds cyclic dinucleotides, means the binding site is large. It's also highly polar, and this large-scale conformational change that's needed in order to activate. So we're not just looking for binders here, but we're looking for small molecules that stabilize the active conformation of the structure and therefore induce signaling. So now I'm going to show another molecular simulation movie, which is a compilation of hundreds, if not thousands, of simulations that we ran over the course of this project to get to our clinical candidate. So again, we're looking at all atom molecular dynamics simulations, treating the system as accurately as possible with respect to the biological conditions. If we just strip away the waters for visualization purposes, we could first see this large conformational change, about 20 angstroms, latching around the cyclic dinucleotide. So our goal was to say, can we induce this conformation of the protein, but without a cyclic dinucleotide, with a small molecule with good small molecule-like properties? The idea we came up with was to get a small molecule that dimerizes on itself in the context of the binding site. Now, this was initially just an idea, but by running a bunch of simulations, we were able to determine there was a high-energy water patch here, shown in red. So we assessed the water thermodynamics, and by building out there, we got some binders. We then optimize this pi-pi stacking interaction between the molecule and itself, and we developed a novel quantum mechanical method really in real time in the project, and that's a lot of what we do, is develop tools based on needs, and we optimize the pi-pi stacking here to really stabilize the protein, and that's where we got our first signal molecules with cellular signals. We then further tried to optimize the potency by building further out into this high energy water patch. And we started to see some very interesting behavior here. For example, in this particular case, we see a large scale conformational change of a loop that induced a tighter binding state, but actually a worse signaling state. So with that, we had to take a step back, rethink the problem. And again, we're not just driving on binding affinity, but a conformational state. Went through various hypotheses, re-engineered our molecules, and ultimately converged on molecules that would induce a very specific conformational state that resulted in the high interferon signaling that we're looking for. In addition to all these simulations, we of course had the whole assay funnel built up and a very strong crystallography platform where we solved about 25 crystal structures down to about 1.5 angstroms in resolution. So we have a high degree of confidence in these structures and in our predictions. Just a little bit more detail about some of the thermodynamics, for example. Here's a close-up of those high-energy waters, and you could see how this type of approach really can reveal something extremely insightful in terms of design. And so the initial idea itself was shown here back in yellow in this top part of the figure, and we could look at all the waters. This is a water thermodynamic analysis where green means that these are roughly equioenergetic with bulk solvent. Yellow means they're somewhat high energy, and red means very high energy water. So these waters are highly constrained. They're entropically very unfavorable. They're very unhappy to be there. They're there because it's better to have water there than have a vacuum. Nature does not like vacuums, but it's better to put a ligand in there itself. And so by putting a ligand into this spot with the high energy waters, we displace those waters, liberate them into bulk solution, and gain a high amount of entropy. And that's what led to our initial binders. After binding, we started looking at stabilization, and the plot in the top left here, we could see there's actually not a lot of correlation between binding alone and the interferon signaling that we're looking for in the cell on the x-axis. And so what we did find is that going beyond binding and look at thermal stabilization, this is DSF, looking at the delta TM, we found actually a very nice correlation between that and our interferon signaling that we're looking for. And so we actually started using that as our frontline assay. I should also mention that it's extremely important to have broad isotype selectivity. And so in addition to just going after the wild type form of STING, we went after the highly prevalent HAQ allele, as well as the relevant efficacy in tox animal models. And as we could see here, our molecule SNX281 in cyan, it stabilizes all of the relevant forms of the protein, similar to the endogenous cyclic dinucleotide in terms of stabilization, but much better drug-like properties, whereas the cyclic dinucleotides have almost no half-life in plasma. Our molecules are quite stable. And we could see this in contrast to DMXAA, which was an early compound that was explored for STING, and we could see that it stabilizes the mouse form of the protein, but has almost no stability of the human form, and therefore no real clinical relevance. I mentioned also that we optimized the pi-pi stacking of our molecule on itself. Pi-pi stacking is an unintuitive type of behavior that we can't really just use traditional med chem intuition to design around. And this case is even harder because we're not modifying one molecule with a fixed side chain, for example, of phenylalanine, but we're modifying the two parts of the molecule together because this is a dimer. So anytime we make a change to one copy of the dimer, one copy of the monomer, the other copy is changed as well. In order to assess this, we developed this quantum mechanical method called FSAP. That's a fragment-based or a functional group-based symmetry-adapted perturbation theory approach. And we developed this in real time, and that's one of the advantages that we have in having our own development team, having the source code, and building everything from scratch, is that when we encounter a problem on a particular target, we can design and develop the tools to solve that problem, and we could turn that around quickly. And every target these days, because they're interesting and challenging, means they're going to have these unique bottlenecks associated with them. And in this particular case, this dimerization was critical, and there are just no tools that exist to solve this problem, because this has not been, there's nothing like this in the PDB. So there's no reason for tools to have been developed. So when we encounter these problems, we develop the tools necessary based on quantum mechanics and statistical thermodynamics to directly build out these in silico assays to solve those problems. Another one of our in silico assays is our binding free energy calculations. We call it RBFE, relative binding free energies, because we start with a lead compound and then make modifications to it, as you typically do in hit to lead and lead optimization. And as we can see here, our predictions are quite strong. We look at the distribution in blue here are the molecules that we say are better than a reference molecule. In red are the molecules we say that are worse, and we could see that the average binding energy of the molecules that we say are better tends to be better than the starting point of 0, and the molecules that are worse tend to be worse. So you clearly want to be selecting from this blue distribution. Now, predictions aren't perfect. All models are models. They have value, but they're not perfect. And in this case, we could see that this model is particularly good and helped us drive to our clinical candidate on a really hard target, STING, with less than 200 compounds synthesized in total. Another key tool that we've developed and are continuing to develop is the integration of experimental data into our simulation data. And this is some work, very nice work from our CTO, Huafeng Zhu, that's referenced here down at the bottom, where we've figured out a way to include biophysical data, NMR data, SAX data, hydrogen deuterium exchange data, for example, into our molecular dynamics simulations. So let me just pause for a drink real quick. So with all of that, we bring this together in our design meetings, where we put experienced medicinal chemists together with modelers and simulation experts. We use experience in addition to water thermodynamics, atomic energy decomposition, these molecular dynamics simulations with conformational modulation, and all of this with a customized visualization interface that we've developed to try to convey as rich of an experience as possible to the design team so they can understand how the system works. Where are the high energy waters? Where are the hot spots? Where are the flexible parts of the protein? Where can things change? And based on this, the design team comes up with their ideas, typically hundreds of ideas on a weekly basis that we run through our in silico assays, our binding free energy assays and conformational energies, in order to filter down from these hundreds of ideas to a handful of ideas that we send out for synthesis or do the synthesis internally and iterate on this process. And so we've really optimized this and feel that it's one of our greatest strengths is this ability to bring the computation into the mix from the beginning and throughout the whole process while still having very strong experimental capabilities as one needs for drug discovery. What came out of this whole process on STING was these small molecules, what we call SNX281, that's very potent in terms of both the wild type and the HAQ allele. And we could see that we get much stronger activation than the cyclic dinucleotides, our compound shown here in blue, the cyclic dinucleotides shown here in orange. And ultimately, this leads to complete tumor regression after a single dose in CT26 PD-1 resistant tumors. And this is shown here on the left side. This is a cohort of N of 8, and you could see, you can't even see the error bars. They're buried under the points because it was the same in all cases. A single dose of SNX281 results in complete tumor regression, as shown here in the cyan line. Very interestingly, what you see on the right here is that there's complete immunity when re-challenged in the same cell line. So if you take the same CT26 PD-1 resistant tumors in a mouse, you insert those into the mice that have already been treated and cured on the left side, and all 8 in this particular case survived. You reinsert those tumors, and without any additional administration of SNX281, the tumors do not grow at all. So we feel quite excited and happy about this project. It was a really great way to get started, extremely hard target, and we were able to advance this very quickly to the clinic using our tools. So I want to spend the last few minutes talking about our target selection process and a methodology that we've developed called conformational genetics that helps us really understand the relationship between human disease-driving genetics and disease and how to treat the disease. So we start with the human disease genetics, as I mentioned, looking at for gain of function or loss of function mutations and associations with diseases. We map those onto 3D structures, and from there we could start to understand druggability and dynamics of the system, which then guides our target selection and design strategy. As one example of this, we've looked at SHP2, and what you see here in this case is that there are these hotspots that are mutations associated with gain of function. On the left, you see some in the SH2 domain. In the right, you see some in the phosphatase domain. And the question is, how are these related? When you map them onto the structure, you see that they're all right at the interface here between the SH2 domain and the phosphatase domain. So that gives a clue that they are related in some way. But how can we actually understand that? Well, we understand that by actually introducing those mutations into a structure and running conformational free energy simulations where we actually pull on the system in different ways to try to see how well those mutations stabilize or destabilize the system. And based on that, we can quantify how stable that structure is. So here we're just going to look at one movie showing the dynamics there. If it plays, it looks like it might be frozen here, so we might have to pause for a second to see if it plays. But we're looking at the dynamics here where the green domain actually will come off of the purple domain here, and that's the SH2 domain there. And what we can do is we can measure this in the context of wild type, as well as the various gain of function mutants. And that, first of all, informs us that our hypothesis is valid and the computational tools can model this. So we see that the wild type, the gain of function mutants show that this SH2 domain, this regulatory domain, comes off easier in the context of the mutants. And then based on that, we could start to drive on different compounds that you could see here that act as glues or stabilizers to hold the thing together. And we were able to very rapidly in this project drive compounds from a relatively poor starting point in a couple iterations to over a log unit of improved potency, both not in terms of just binding, but in terms of stabilization and cellular signal. One more example of this type of approach is shown here in NLRP3, part of the inflammasome. And what you see here on the right is the full inflammasome, this 11-mer that you can imagine if you had to disrupt this, how would you do this with a small molecule? Well, if you look at just one of the monomers of NLRP3, you can see that there's actually quite a interesting conformational change that takes place from the inactive state, shown here on the far left, to the active state on the right, where this purple domain rearranges and gets out of the way, which allows for the formation of the dimer and then ultimately the multimeric 11-mer for the inflammasome. So our idea here is that if we can understand this conformational change, we might be able to lock the protein into an inactive conformation before it even has a chance to form those higher level states. So again, we'll look at a little bit of a movie here. It looks like this one's working. These are the types of simulations we're running where we're seeing quite large conformational changes going back and forth between this active and inactive conformation. This is based on a method that we've developed in-house using metadynamics as well as an adaptive biasing force in order to get these accurate results on these longer timescale large motion events. And what we see here is that this gain of function mutations, these gain of function mutations all reduce the barrier between going from the inactive to active conformation. And so our goal here would then be to see what can we do to lock this protein into this inactive conformation. So we've developed a suite of druggability tools using molecular dynamics simulations, including water thermodynamics, mixed solvent simulations, as well as some other cavity-based searching tools. And we were able to identify a pocket right at the interface of these two domains that looks to be quite druggable. We've ran a series of virtual screening experiments using a methodology that we've developed internally that includes traditional virtual screening up front, but ultimately culminates in high resolution binding, absolute binding free energy calculations with all atom simulations, as we've discussed before here, to make our final selection. And the nice thing about this is it breaks away from the traditional paradigm of rigid receptor docking. We actually allow for full protein flexibility. We account for ligand strain, desolvation, entropy, water, and everything else that really matters in the context of binding. And we've validated this on multiple targets, now even broader than just within the innate immune system. And what we've been able to identify are novel ligands that both bind and appear that they can lock this protein into this inactive conformation by bridging the gap between these two functional domains. So with that, I want to just wrap things up saying that we've accomplished a lot in the 4 years we've been around, and really developed a company that can take things from chip to clinic. We built an industry-leading platform for physics-based and physics-driven drug discovery, coupled with all of the drug discovery infrastructure necessary. We develop custom apps as needed for drugging challenging targets. Some of the key breakthroughs come from this integration, and specifically the integration of biophysical data allows us to access longer timescales that we can't access with traditional simulation methods. And our small molecule STING agonist is really showing the value of this approach. You can learn more on our website, which is updated regularly. We have a YouTube channel with a video that just describes a little bit about what I said and a little bit more of a qualitative and visual representation. We have a great team, great culture, and we're always hiring. So if you're interested, check out our site, learn more. And with that, I'm happy to answer any questions, and thanks for joining.
Thanks, Uri. A great talk. I have to, I started my studies in biophysics and protein folding, doing molecular dynamic simulations when I was a sophomore in college. And it's come such a long way in the last 20 years. What can be done now with high performance computing and GPUs and allowing you to do simulations over these large timescales of different kinds of capturing, the whole plethora of dynamical motions you can have, right, all the way from pico timescales to millisecond timescales. But that's great.
Absolutely.
Wonderful. And we do have two questions for you, Dr. Woody. One is from Miss Janet Pina. She's asking, amazing simulations that streamline the analysis of molecules in their environment. Is there any movement towards VR technology that allows for another view of the data?
Yeah, absolutely. So we're big fans of different ways of looking at data. That includes different ways of plots and visualizations, but a lot of that ends up being these movies and these trajectory visualizations in different modes. And so we are actually exploring that. We actually have been working with Nanome. If anybody's familiar with them, you'll know, and if not, you should look them up. Nanome is a company based in San Diego that is really leading in that area of virtual reality and bringing that to the forefront for drug discovery. And so they have a great platform, a great company. They move very fast, and we are using that internally and developing some apps very specifically around the Nanome VR platform. It's really something worth looking at.
Wonderful. Yeah. And actually, funny you said that, Dr. Woody. Nanome is one of our event sponsors. Their CEO, Edgardo. And I think they might actually be in this room. If you guys
Excellent.
are, please.
Well, I would recommend anybody go check them out at this conference or elsewhere. It's a really nice technology.
Wonderful. And there's one more question from Dr. Steve Miller, CEO of Sandia Biotech. He's asking, brilliant presentation, and I'm a huge fan. Are you interested in combining virtual reality tools into your program that align with your tools?
Yeah, there you go. So same, very, very similar question and same answer. Absolutely. I mean, we're really just interested in not choosing a particular technology or a particular way of doing anything, but thinking about everything on a case-by-case basis and seeing what's the right fit for a particular problem. And I think virtual reality has a lot to offer. The human brain is extremely good at recognizing patterns and seeing things. The more we could present to our brain to use our, all of our senses and pick up on interesting information to make better decisions, we highly value that, and we think there's a huge future there. Again, I think Nanom's done a fantastic job. I'm sure there's other players out there as well in academic endeavors, but getting it right is really hard. It's not just a question of being in virtual reality. Initially, virtual reality, if it's not done right, just feels kind of like a game and it's fun, but doesn't provide much value. But if you do it right, you can really gain a lot of meaningful insight to help drive decisions. And for us in particular, these simulations that we're running, these physics-based simulations, they predict accurate results. They predict accurate numbers. But the number, this molecule is better than that molecule by a kcal per mole is nice. But even more important is, what do you learn from that? What is it that drives this molecule to be better than that molecule? And can we learn from that and come up with a next iteration of design ideas that are even better? And that stuff all comes through, human visualization and intuition. And the more we can bring to the table there, so layering on the physics into the visualizations and the virtual reality, I think is a very fruitful path forward. And I think we will all see this as part of the future of drug discovery.
Wonderful, wonderful. So if there's any other questions for Dr. Woody, again, I will put up his slide deck. If you are interested in learning more, there's the website for Silicon, their official YouTube channel as well. And we do have someone from Nanom, Daniel Gruffit. He said, "Thank you for the shout out, Dr. Woody." So
absolutely, pleasure.
Wonderful. So with that said, we will be heading to our next presentation from Dr. Shruti from Novartis, Dr. Dus. And again, thank you, Dr. Woody, for your presentation.
Yep, thank you, and thank everybody.